Welcome to the STEM Power podcast, where we discuss emerging scientific research with leading scientists. When we think about traumatic injuries to the brain or the spinal cord, it's easy to focus on the immediate physical damage. But what happens afterwards at the cellular level? That either helps or hinders recovery. This is an evolving area of research that has led scientists to shift from a one-size-fits-all solution to a more personalized, multifaceted strategies. So understanding how different types of cells like astrocytes react to injury can help shape better treatments and improve patient outcomes across various disciplines. So joining us today, we have Dr. Ina Wanner, an Associate Research Neuroscientist at the UCLA Geffen School of Medicine. She's part of the Intellectual and Developmental Disabilities Research Center at the Semel Institute for Neuroscience and Human Behavior, and she's been leading research that bridges the gap between disciplines to improve outcomes for patients with traumatic brain and spinal cord injuries. Dr. Wanner has earned a master's in biology and a Ph.D. in human biology, where she discovered key mechanisms like local protein synthesis and brain plasticity. After 2 postdoctoral fellowships, including work at the Miami Project to Cure Paralysis, her research now focuses on neuron-glial interactions for injury regeneration. In today's conversation, we'll dive into how Dr. Wanner's research is reshaping how we diagnose and treat traumatic brain injuries, and we'll explore her recent work on astrocytes, those fascinating supporting cells in the brain, and how they respond to injury, as well as her patented invention of biomarkers that have revolutionized the detection of traumatic brain injuries. And finally, we'll touch on her experience in a multi-site consortium, working collaboratively to tackle these complex issues. So with all of that in mind, I'm very delighted to introduce Dr. Ina Wanner. Good afternoon, Dr. Wanner.
Good afternoon, Simran, and thank you so much for having me today on your very interesting podcast series.
Of course. And without further delay, after giving the introduction, I'm very excited to get started. So, Dr. Wanner, something that I like to ask all of my guests before getting into the details of your paper is to ask what sparked your interest in research. Specifically, your research is in neuroscience and it focuses on traumatic brain injuries. So, where did this interest come in?
Well, first of all, thank you so much for your kind introduction. I have not been all the time interested in a very translational field, which studying injury to the brain is, or to the spinal cord. Or originally, I was just always curious about mechanisms in nature and how things work in nature. And it was not until I actually joined the Miami Project to Cure Paralysis, where I really found this strong mission to help patients that have paralysis to do research that really improves regenerations of fibers. So it allowed me to use my skill set and my curiosity on cellular processes in the central nervous system and really apply it to translational questions, because I met surgeons there on a daily basis, and we had patients in the wheelchair asking us in the hallways, when are you curing paralysis? And so to contribute with essential questions was an eye-opening and a transformative experience for me.
It's always so nice to hear that the guests that I'm interviewing and your research is really based on these experiences that you've had with people. I really liked how you mentioned that you were seeing these patients in the hospitals directly asking you, when are you going to find this answer for me? I'm sure that was probably a pretty pivotal moment in your research career. So it's really great that your drive is really towards helping people. So, Now, I want to ask you about some of your research. I've read online that the Wanner lab uses a trauma model to reveal how brain cells respond to injury. And this trauma model is meant to model whiplash trauma in the brain. And really, what your research did is that one aspect was investigating how changes in protein levels correlate with stages of cellular damage, specifically in astrocytes, which are a type of brain cell. Now, this sounds really incredible, and I'm specifically curious about this model that you created, this in vitro model. So, can you explain to us what consisted of this human trauma model a little bit, and why were astrocytes specifically of interest to you?
Certainly. Our human trauma model, we are using donated human brain cells. And I want to first put in a huge plug for our ethically mindful regulatory way of procuring the tissue. We receive it from pathology. Patients who undergo elective abortions here in California at UCLA are asked if they would like to donate the remains, and we are not interacting with patients or with the physician, so there's no incentivizing, and it is following all ethical regulations of privacy and regulation. So these tissue remains from that include the fetal brain are then shelved in our pathology core lab, where we then receive the tissue, we isolate the astrocytes, and that is one of the reasons we're working with these glial cells of the brain, because we can actually expand them into millions of cells, and that allows us to do these sort of very standardized experiments that allows to address studies that require high throughput for drug testing, for instance, as well as proteomic studies that require milligrams of proteins. And it's translationally relevant because we're really investigating how the biomarkers are released from actual human primary cells that are differentiated in a dish. And here is the model. We're using actually culture plates that have a rubber membrane, a elastic membrane. And then when the cells are mature, they receive an abrupt air pulse that is actually deforming and stretching the cells, which then show features of cell wounding that occur just in the brain at like interfaces between white and gray matter that has been shown. And so this mechanical trigger, this stretch, is a model that models concussion and brain injuries, but it's controlled, and we can study therefore better how drugs respond to how cells respond, how drugs can be used, and what markers are released.
In vitro modeling and any type of modeling, really like what you're doing here, is always so interesting to hear about. I work in a lab as well, and we do in vitro modeling of the eye. And it's always really cool to hear about this modeling of organs and of different parts of our body that are more within, and to see how you can do that type of modeling, but within a controlled environment. So it's really interesting to hear about how you guys were able to create this mechanism to, I guess, model all of these processes that happen in the brain. And after doing some more reading about your research, we learned that this research itself has led to patented astrocyte biomarkers for traumatic brain injury diagnosis and monitoring, which is just so cool, especially since we have not yet had a guest on our podcast that has patented their research. So I'm really curious about some of the next questions that I'm going to ask. But before we get into those, I want to get the listeners on the same page as us. So can you first explain what problem did you aim to solve with your invention or your discovery?
So actually, we discovered this trauma-released proteins or proteome that we call traumatome by accident. It was when I worked initially at the Miami project to cure paralysis, we tried to model what those astrocytes are doing when they respond to injury. And so they're forming borders to protect the remaining nervous tissue, the remaining spinal cord. And so they make scars. So we use this model as like mini scars in a dish. And when we actually looked into all the proteins that these astrocytes change and secrete, we actually identified that these were proteins that were inside the cells. So this is actually a wounding response that we have stumbled on, and then we thought, got lemons, make lemonade. So we basically decided to pursue this idea of, hey, when the cells are wounded and release all these intracellular proteins, can we capitalize on that, and can we maybe identify markers that point to the brain that are solely in these astrocytes, because they are actually sitting right at the blood-brain barrier. And when there is a concussion or brain injury, they are the ones closest to releasing proteins into the bloodstream. And so it was fairly easy to initially say, okay, these are all the proteins released. We call them traumatome. And it was a much, much harder step than to identify individual potential biomarkers for brain injury, which we have done in the years since around 2014. And then, of course, at that time in 2014 was a big question: is concussion real? Are the chronic effects of neurodegenerative diseases that athletes and football players or boxers experience, are these real? And the National Football League in America has been denying this at the time, even though pathologists have pointed at brains after brains from athletes who donated their brains, showing these neurodegenerative changes that prematurely developed in these athletes. So at the time, the big question was, can we develop an objective blood test that will unequivocally inform us of a brain injury early on, before all these degenerative changes occur in a person?
Right. And you spoke about it a little bit already with the biomarkers and this blood test, but can you kind of explain in the most simple way possible, what exactly is it that you invented or did you find with answering this question about the blood test?
So when we looked in more detail on the amounts and the kinds of proteins that were released, we actually found that early on, in the minutes and hours after a trauma, the cells release markers or proteins that are typical for membrane damage. So when the cell is wounded, we have a typical signature for cell wounding. And our human brain cells, they take a long time before they undergo cell death. This was another discovery that is not the case in mouse or rat brain cells, because their metabolism is much faster. But in the human brain and in our model, the cells take a long time before undergoing cell death. And we see this in trauma patients, that there is a prolonged time before there is neurodegeneration and atrophy. So we've identified markers that actually label or that identify cells that undergo cell death, and markers that are indicative for the early wounding event. And so these are then, we went to really focus on those markers that point to the brain, that are brain specific, but very abundant. And those markers like the aldolase C or a brain lipid protein, they are so substantially released that we can detect them in the blood very early on. And so they're substantially enough released to also serve as a marker for milder injuries, where, like concussion, where there's only cell wounding. And that is really the distinction between the markers that are presently generally being used also for neurodegeneration, which are more identifying dying cells.
Right. I really like how practical this is to being able to detect these specific biomarkers and everything just through a simple blood test. It's definitely something that can be applied very readily in these mainstream sports, like you were saying, boxing and football, where players are getting concussions and brain injury ever so often. Now, my next question isn't super technical. It's just out of curiosity. Patenting something, as you've mentioned, can take a really long time, not just for the logistics, but for the research itself. So I'm wondering, how long did this take, kind of from the beginning of the research up until the point where it was patented? How long about did this take for you?
First of all, let me tell you that we were very fortunate that this is not an endeavor that my lab and my collaborators had to do by themselves. We partnered with our licensing partners, the company Brainbox Solutions, and the CSO there, Tim van Meter, worked rigorously and tirelessly to make a reproducible, robust blood test. So that's the end, and that is about 10 years after the original invention report that we filed, where we identified a multitude of proteins released after trauma. So, a long story short, it requires specific tools, these antibodies, and it requires actually two of them to sandwich the biomarker to basically measure it in the bloodstream sensitively and specifically. And on the other side is a long process that is a legal process where in each country separately, the biomarker is scrutinized that it is really novel and that it is a unique different approach in measuring or in detecting brain injury or concussions than what is already being patented. So at the end, we have at the moment two patents that describe a various combination of markers in combination of uses in Israel, in Japan, in America, and other countries like Europe are also approving this patent.
Right. And along this whole process, specifically in the lab, like you were mentioning, with detecting these biomarkers and finding the ones that can target specific properties of traumatic brain injury, did you face any significant challenges along the way with this research? And if you did, how did you overcome them?
Yes, absolutely. So there were several challenges. One challenge is really to find those tools, those antibodies to make a specific blood test. I already mentioned that. Another challenge at the time was that the funding bodies were still in denial, and so it was hard to get funding for it. However, we have been fortunate to pair up with diagnostic companies, and the largest of the companies actually asked us to remain in secrecy about our discoveries, which was a huge challenge as a scientist, as a researcher. I always want to share my data in scientific meetings and symposia. And so to be sort of literally shadowed by a diagnostic representative from the diagnostic company was a real challenge to not be able to present my work. However, nowadays, there is a different climate, and the diagnostic companies in the field are actually welcoming sharing and communication and being transparent with whom they work with. So that is not a problem with our current partners, Brainbox Solutions, who we actually sponsor those events where biomarker data are exchanged, which is a better way forward. Other challenges, of course, are from competitors. But I am a very collaborative person, and my philosophy is always scientists and researchers are curious, and it's better to collaborate than be competitive.
Of course. I think that's a really good takeaway, actually, to work with other people and to get ideas from other labs, because that can help you grow and help grow your research as well. And I was going to ask these next few questions later, actually, but I think right now is a good time to ask them. You work within a multi-site consortium, those types of studies, so within or with a bunch of other labs. Can you explain kind of what you do with that? What's the process like? Just a bit about that.
Thank you. Yes, we were basically tasked to use multiple non-invasive assessment tools in preclinical research, in rat models of brain injury by the National Institutes of Health, because the two fields, preclinical and clinical, are still using very different approaches. And this is one of the reasons the NIH and the National Institutes of the Neurological Disorders and Strokes identified this as one gap of translation that is holding us back and that has contributed to the failure of so many translational ideas that of so many treatment strategies that work in animal models and not in the clinic. So we were tasked to implement some of these biomarkers in neuroimaging, and we were also tasked to do this in different institutions and exchange our experience and exchange how we do things so that we can, similarly as done in a clinical trial, that we can actually standardize our approaches between labs. And that is what we have been doing for the last 5 years. 7 years actually already. It was a
Wow.
5-year funding, and we are still working on analyzing a lot of the data.
Wow, it's been a really long time then, I guess. And I'm wondering really, because you mentioned you're working with different institutions, and you're trying to maintain consistency with these studies, have you seen any major differences between running the same experiments but in different institutions? And how do you go about maintaining that consistency?
Yes, that was one of our major findings, that brain injury done on rats in Johns Hopkins University or Georgetown or UCLA actually turn out different. And so although we have tried to have all our settings standardized, however, we have been finding that this is, of course, one of the clinical practice that physicians are seeing patients with a whole different range of injuries. That is one of the features of trauma to the brain. It is very heterogeneous, and no one patient is like another. And so how we are addressing this is by standardizing our assessment tools, because here is what we can do. We can exchange our protocols on measuring biomarkers, and that's what we have been doing. We have been standardizing how MRI imaging is done as much as possible. And we have, importantly, identified and implemented quality controls so that whatever differences in our measurements remain, we can at least put a gauge on and say, okay, this is our variance, and this is how we can assure and express how precise our tools are. And so then by embracing the range of injury severities, we can speak to them by comparing our measurements, because those we can control.
Exactly. I like how you mentioned that, how initially the problem kind of was there was such a big range of differences between the institutions, but then you chose to embrace it more and realize that, again, like you mentioned, not all patients are the same. There will be a vast diversity with the patients coming in. So you were able to use that kind of to your advantage. And while on the topic of this study, this multi-site study, can you share any of the major or promising advancements that you found with this research, if possible?
Thank you. Yes. So we are still in the process of analyzing a lot of the data from multiple sites. But one of the most rewarding experience, I could say, is that we have been trying to really arrive at a way of communicating between different subject matter experts, between radiologists who have a specific approach to their MRI imaging and biomarker experts and people like us doing also histopathology, to really align different findings and then come to a conclusive picture of individuals' rats injuries. And one smaller study that we've done in the first couple of years was very rewarding, where we used a multivariable advanced analytics statistical approach that was together UCLA with UCSF, and the postdoctorate researcher there, Hannah Radebo, and us, we were able to identify using multiple informations and variables from MRI and blood biomarkers and tissue changes that there is injury phenotypes that point to an overall change in the white matter or in the gray matter, or an overall change due to swelling, early edema. And that was very rewarding to see that all these different measurements point to one particular injury phenotype. And we're in the process of writing that up.
That's really interesting. I'll make sure I keep my eye out when that paper gets published so that I can read more about that. And I want to actually jump back to the biomarkers, because you and your team and everyone that you had worked with, you were able to discover these biomarkers that can help detect traumatic brain injury. And specifically, while reading these different papers, there was this mentioning of serum biomarkers. Can you kind of explain, again, in a simplistic way, what is a biomarker, what are serum biomarkers, and how can they help specifically with diagnosing concussions or brain injury?
So a biomarker is a protein that is elevated in the circulation after a brain injury or a concussion that is normally at lower levels or absent. So it needs to be a protein from the brain that cannot be similar to any other proteins of other organs that are undergoing constant rejuvenation, which the brain is not. And therefore, it's pointing to the brain as being injured. A biomarker is also a diagnostic indicator. Concussions in particular are known to be very hard to diagnose, because a person who has received a hit to the brain can have symptoms that are very general. And a biomarker could be an objective and unequivocal measure that there is brain cell wounding. So it will help to diagnose and help early on, as opposed to in hindsight. So it's very important then for deciding in the emergency room, for instance, does this person need further monitoring, further hospitalization, or can the person be just let go and with some advice for rest? So a biomarker can be an objective and early indicator for helping in that decision making.
Mm-hmm. And these biomarkers, like you mentioned, can be detected through blood tests. But in this paper that was talking about the biomarkers that you found, I don't have the exact title of it on the top of my head right now, but it did mention imaging techniques. Can you kind of talk about where did imaging techniques come into the framework of this research with the biomarkers, since they were detected through blood testing?
So our initial findings were in the lab and with this culture trauma model, where we used microscopic imaging to follow the cell wounding of the trauma that was caused by the trauma. it helped us to relate the protein release into the fluid with the cell injury state, the wounding state versus the dying of the cells. And in a more general way, when we then have been conducting studies in animals, we used MRI imaging alongside with microscopic imaging to get at the picture of brain injury in a contusion in the rat, and to provide the imaging underpinnings what is happening at the cellular level by looking at histological sections. At the same time, in the same animals, blood was drawn, and so the biomarker levels could then be confirmed and compared to the underpinnings of changes in the tissues and MRI pictures.
Right. Okay. Yeah, that definitely makes sense. Within the framework, it would definitely help with making those comparisons and then being able to further analyze your data. So, Dr. Wanner, my next question for you is, what are the key challenges that you've seen with multimodal biomarkers?
So, multimodal assessment tools is when in the clinic or in research, multiple assessments, multiple biomarker types, and multiple biomarkers themselves are being combined to gain a further insight into the patient's physiological state after a traumatic brain injury. That can include MRI neuroimaging, that can include blood biomarkers or EEG reading. And this is an important point because it really helps if we have different types of tools to corroborate each other, to give a fuller and a more complete picture of the patient at any time. And the biggest challenge here is twofold. One, the different experts in the room have to actually speak with one another and have to actually express their way of measuring and their findings and their levels of change or whatever their outcome is in a way that everybody else understands it. And so in our consortium, that has been a huge challenge. And so the language needs to be tackled first. And this is a very general, important way of addressing research. Nowadays, we are moving beyond boundaries of individual subject matter fields like MRI or histology or blood biomarkers, and to develop a common language to really make the effort to speak in general terms has been is really critical for if we want to succeed in neuroscience and specifically in the field of neurotrauma. And that's why we have created this more general terms like a data dictionary, common elements of data and of communication, what they mean. And it is only secondary that we need to deal with the actual strategic computational challenge of different data types. Again, if the statisticians and the computer sciences don't understand the same language as a neurologist and a physiologist, again, it boils down to communication between experts to actually proceed on this quest, which really will improve understanding the traumatic brain injury and each individual patient.
Mm-hmm. I really like that you mentioned that, it's a big concern or it's a big point here where you need to have the two different specialists speaking with one another in a common language, no specific jargon. It needs to be very clear because this is really when some small changes or pivotal points can be made when everyone is on the same page and understands really what is going on. So it's really good that you mentioned that. Okay, Dr. Wanner. So now I want to touch more on the longitudinal side of these studies, because as you've mentioned in your papers and previously, that is really the crucial time when this monitoring needs to happen and when your research really applies. So can you elaborate on the importance of longitudinal studies in understanding traumatic brain injury biomarkers?
Yes, certainly. So obviously, the traumatic impact is a trigger for a prolonged process of injury and inflammation and wounding that each individual patient is undergoing this path on recovery. And that really is this time period that determines the outcome. And obviously, physicians and intensivists need to monitor each patient during this course, and it's only the more severe patients that can receive a probe in the brain to actually monitor of various gases, oxygen, CO2, and the pressures of the brain. And it is much desired for physicians and to have non-invasive monitoring tools like biomarkers that a broader range of patients can be better monitored besides those that undergo surgery and have an open skull. In this process, monitoring using biomarkers, these markers are actually elevated on the cusp of any adverse, dangerous pathophysiological event that occurs, like an elevation of the intracranial pressure or a bleeding or a secondary ischemic event like a mini stroke or an epileptic seizure that could happen. And so these non-invasive measures are generally recording not very sensitively, but a biomarker would be actually, so we have measured biomarkers over the course of, say, 2 or 3 weeks and seen elevation of astrocyte biomarkers in patients' cerebrospinal fluid, for instance, early, right before or at the beginning of an adverse secondary event. And so if monitoring with blood biomarkers or CSF biomarkers could be going on in parallel to other monitoring tools in the neuro ICU, for instance, then physicians and intensivists could respond more readily and avert some of those very dangerous secondary events. To do this, we also need to know the kinetic of the biomarkers. And we have done a little bit of research showing that these wounding markers are very fast released. One of our markers is very stable in the blood. Other markers have a very short half-life and are cleared in the bloodstream very fast. And some other markers, like those derived from neurons, for instance, appear not until later or during chronic times. So it is a combination of having biomarker knowledge on their profiles and having the convenience to actually look at progression in the patients and be able to respond in a more timely fashion to improve recovery.
Right. And I like how you mentioned that, this helps with monitoring the progression. And this is kind of where that one size fits all does not fit in, because it helps you to make sure that you're tailoring all of your treatments to your patients, specifically to their needs. So that's why your research was just really incredible to me, because it's so translational and it's so applicable, especially in hospitals, with the people in need. So that was really all of the main technical questions that I had for you, Dr. Wanner, today regarding your research. So as we wrap up our conversation today, I'd love to hear about a moment in your research career that has been particularly meaningful or impactful for you. So could you share maybe a memorable experience or a story that you might have that helped shape your journey as a scientist?
Well, in brief, never give up. I have reached walls, boundaries, people who really are disbelieving. And basically, I'm a very driven person and I never give up, and always pushing forward against boundaries. And this has been also a characteristic feature as a mentor, as I am guiding and mentoring over 100 undergraduate students that I have been guiding in their research projects. And it has been such a rewarding experience for me at UCLA, as well as in Miami and even in Germany as a graduate student, to really mentor students and to show them that they can be very strong if they believe in their project. And this confidence gives them grit and perseverance to overcome all obstacles. And it doesn't require a person to tell them, you're good and you're great. It is something that comes from within. And I have seen several young women and men who have been extremely timid and not outspoken and at times despairing with their assignments. And I keep telling them that they can do it and that this is a particularly important question to address and how they can do it, and then let them do it. And then I have seen people flourish and become very confident and then training others in the same manner. So it has been an incredible journey to see students really take charge and become the best and confident person they could be. And that could not have happened if they would have not engaged into their independent own research project that has given them all that empowerment. And that will not go away. It's not like a surrogate that basically another person have to keep patting them on their back. They are on and going and running, and they do so independent of whatever comes. And so this has been one of my most rewarding experiences, seeing that happen with Annika Myers, with Salma Arredondo, with numerous other students who have basically surpassed their own limits.
I love that you mentioned some of your students as well. It's really inspirational to hear that from you as well, such an esteemed researcher and scientist. And it's a great reminder to give to listeners and to everyone that really it is very important to never give up and to believe in whatever your research is or whatever your project is, like you mentioned. So thank you so much for sharing that delightful experience, rewarding experience, Dr. Wanner. I had a lot of fun today through our conversation and learned so much. So just wanted to thank you again for taking the time out of your day today to be on STEM Power and speak about your expertise. It's been a pleasure.
Oh yeah, I'm very grateful for you reaching out to me, and thank you so much for preparing this interview and giving me the chance to share a little bit of our research. And good luck with your podcast, and I look forward to other sessions.